Hi, Dr. Bordi.
Hi,
I
can you see my full screen?
can see your screen.
Awesome. So, thanks for inviting me here. It's always a very good opportunity also because from this symposium, there is a lot of interaction between different groups on the world. So I had the pleasure to interact with some of the guys that they presented their own results, and I guess this is very, very important because, we try to summarize our experience and share all the results beyond this symposium that, sometimes can be a little bit boring because uh, we explain too much and we have so many details that for us are critical, but I see that maybe for most of the, let me say, common people, it's very hard to get to grab the real message.
Yes.
So
Next year we'll have dance breaks.
yeah, so I try to simplify a little bit my message, even if I'm gonna present you now the full story that we recently published less than 1 month ago. And this was a big effort. I skipped the ADSL. So in this recent publication, we focus our attention on mitochondria. So we move our attention a little bit far from what has been described so far on ADSL deficiency. And we, let's say, focus more on metabolism in all the possible way. So this is a table that summarizes all the cells that we use because we experience a lot of troubles when we also try to publish this story because one of the major problems that we occur was the fact that let's say normally when we try to publish a story, we send the story to a journal and we receive some comments by referees that are unknown, of course. And in some cases, especially in this case that ADSL deficiency is a very rare disease, it's going to be complicated to, let's say, provide a lot of, let's say, data from different patients because this is something that normally is done a lot in cancer. Then normally we have thousands of patients, or in other diseases where it's very, very common to have at least 20, 30, also 100 different patients that can, let's say, straight the message, but also the results. In this case, as you know, it was a little bit more complicated, but we tried to cover, thanks also to Marie Zikanova, to cover all the possible, let's say, type of ADSL deficiency disease. And we play in a different way. So because as we noticed, there is a lot of, even if we are talking about the same disease, but the moment that we move and we use models derived from patients, each patient is almost unique because in this case, there are different mutations, and we realize that this can cause a sort of mess, literally, because it's difficult to understand what can be common between the different model that we use and we derive from each single patient, and what instead is specific of that patient, okay? And this in science is, let's say, create a sort of foggy message because sometimes we should trust on our results and we have to repeat the experiments over and over to figure out what is due to our hands because we can, of course, make mistakes, and something that instead can be just related to the specificity of each single patient. So we derive different models. We play with the blood cells and we're able to derive what we call immortalized lymphoblastoid cells. We derive cells from the skin that we call fibroblasts. And we call primary fibroblasts because we didn't touch. We just derived the cells from the skin biopsy and we use for our experiment. And then, because we are molecular biologists, we try to derive some other model that is named IPS cells. basically we are able to reprogram the fibroblast in order to get some cells from which we are able to obtain neurons or muscle fibers or other different kind of cells depend on specific protocols that we use in the lab. So let's say that these IPS are more, let's say, are very common now because there are a lot of different ways to use these cells, and because we can derive, for example, neurons that and in which we can figure out what's going on at the molecular level. Okay, and these models are very useful. And normally, what we try to more or less, sometimes we have troubles because some of these models are very expensive just for the generation of these cells. We try to repeat the experiment in all these models in order to, say, get the more or less similar results and to reach a point that could be relevant for the understanding of the disease. So something that I would like to point out that was unexpected was one of the first results that we obtained the moment that we start to analyze these cells derived from patients. So again, fibroblasts are derived from skin biopsies. Instead, EBV, LCL are cells that are immortalized and are derived from the blood samples, so from B cells that are part of the immune system. And in all the cases, what we notice is that in ADSL patient, there is a decrease of ADSL protein levels, but also the mRNA level is a significant decrease. This means that even if we are talking about different mutation and a point mutation, these mutations are able to affect the amount of ADSL protein. And I guess this can be very relevant because we are not just talking about a specific mutation that can affect the function of the enzyme, but this mutation can also affect how the ADSL gene is activated. And this means that there is also a defect in the protein level. So meaning that this mutation can affect ADSL in all the possible way. That was completely unexpected because in most of the cases, when we talk about rare genetic disease where there is just one single point mutation, is very unlikely that you have this kind of defect. And it seems that this defect is let's say, linked to the severity of the disease. So means that, let's say, when we talk about mild and severe disease, we observe a stronger downregulation of DSL protein and RNA. So probably there is a sort of feedback that can explain this alteration. And now we are working to understand and figure out why this is occurring and probably which is the molecular mechanism. But in this case, our first goal was to figure out, okay, so if a single point mutation can affect the protein level, so there are two different options normally. Or the protein per se is degraded faster, so means that there is a faster turnover. So proteins are like a car. So if I drive the car for long distance, of course, I need to make a revision. So refill the oil, do some maintenance to keep my car working. And proteins are like this. So let's say in the moment that we observe a decrease in the protein level, probably our idea was that this decrease could be related to an increase in the degradation of this protein. But it's not the case. And we excluded the decrease of the ADSL protein level could be related to an increase of the degradation of the protein, but also on the stability of the protein. So this here we use different drugs in order to study this, and we exclude that the defect in the at the protein level of ADSL in ADSL deficiency patient cells are linked to increase of degradation or decrease of the stability. Okay. And then because I mean, I'm a biochemistry, so I work a lot of metabolism. So we try to figure it out if there could be a way to, let's say, discover different function of ADSL, because normally when you see a defect, for defect, I mean, these cells per se grow slower. So sometimes they are bigger in terms of size. So the morphology is a little bit affected. So rather than focusing on, let's say, common pathway, we try to escape a little bit, let me say this, from this common view and try to figure out if there could be other, let's say, way to explain the pathology. Because, of course, the nervous system is the most affected, let's say, system in this disease together with muscle. And because these two, let's say, system relies a lot of mitochondrial metabolism, we try to dig a little bit around to see if ADSL mutation could, let's say, be linked to alteration of metabolism. The idea was metabolism in general. Then we did some bioinformatic analysis, and we found out a link between ADSL and mitochondrial metabolism. So mitochondrial is the, let's say, the power hub of our cells. It's famous because let's say, we describe the mitochondria as the most important, let's say, area when ATP, so energy, is produced and, let's say, is being related to ATP production. It's not like just like this, because mitochondrion per se is important also to regulate a lot of different aspects of cell life, from, let's say, not just energy, but iron level is important to the production of urea, is important for amino acid metabolism. So let's say that is the metabolic hub of the cell. If there is a defect on the mitochondrion, you're going to have a lot of troubles around in the cell, okay? And when I say in the cell, means that also the cell cannot, let's say, properly communicate to each other, okay? Or reply to external stimuli. So based on this connection that was due by, and we found out by bioinformatic analysis, so using different tools rather than working on the bench of our lab, we moved back to the lab and we start to do some very common way to we observe the morphology of the mitochondria, because mitochondria are very, let's say, beautiful organelles. So they are not static. They are, in most of the cases, if you look around and you search for mitochondria, what we found on internet, these mitochondria look like a big bin, okay? But they're not like this. So they are not static. They can be elongated, they can move around, so they can be fragmented because they need to be transported in specific part of the neurons, for example. They need to, let's say, move properly. And they are very dynamic. But the moment that there is a defect on the mitochondria, so what we observe normally, and something that I experience also when I work on Down syndrome, that there is a defect in the morphology. And for defect, I mean, there is a fragmentation. So, and as you can see from these pictures, looks like that this fragmentation is more evident when the disease is more severe. So in the severe and fatal neonatal form, these cells, they show mitochondria completely fragmented, while in the very mild that the mitochondria looks more or less healthy. But if you see in the control cell, you have this nice spread all over of these mitochondria that they look like a big spaghetti. Let me say this because I'm Italian. And and this is the most, you say, the easier way to describe how the mitochondria look like. So based on the fact that there is a defect in the morphology of the mitochondria, okay, what's going next? Let's see if there is a defect also in the activity of these mitochondria. And so we use different analysis, and we prove that not in the very mild cells, but only in the mild. We confirm also in the severe cells. We couldn't analyze all these cell lines because there were some troubles. As I said, the cell derived from, let's say, severe patient or fatal neonatal patient, this cell, they cannot grow properly. So it was very hard for us to, let's say, make experiment on these cells. So, but let me say that in the mild cells, there is a decrease in the mitochondrial respiration. And this defect is compensated a little bit by the increase of glycolysis. So the cell can feel that there is something wrong on the mitochondria, and probably they try to compensate, they try to make energy, derived ATP from the glucose directly, upregulating glycolysis. And this, again, something that we also see around in a lot of the cases. The critical point here that not only the mitochondria are more fragmented, not only they, let's say, work less, but is also a defect in terms of the number of mitochondrial DNA copy per cell. What does it mean? That the mitochondria, they have their own genome that we call mitochondrial DNA. And this is relevant because on this small amount of DNA inside the mitochondria, there are some genes that are fundamental for the activity of mitochondria in order to, let's say, make oxidative phosphorylation. So it's the way how the mitochondria can, let's say, make ATP and thus, let's say, store energy on the cells. Okay. So we did some experiment also on iPS cells. So this, when we reprogram it, so reconvert these fibroblasts into something else in these cells that we call pluripotency cell lines, because we are able to differentiate these iPS cells in neurons, as I said. And we confirm that also when we try to do this in those cells derived from ADSL patients, there was a defect in the maturation of these cells into neurons, meaning that the mitochondrial defect is occurring also in these cells, and probably this defect can drive, a bigger alteration in the, let's say, propensity of this cell to become neurons. And this is critical, especially in the first stage of development, okay, when, let's say, the nervous system take place. And we confirm that there is alteration of mitochondria also in these neurons. I'll try to summarize a little bit all this graph. And then, because at that time, when we start to work on ADSL deficiency, we were not able to work on C. elegans. So we decided to use a different model that is the fruit fly. So the Drosophila melanogaster, that is very common around, is also another animal that is largely used because as for the C. elegans, we know a lot on this animal. Second, because it's very cheap to work with. And the critical point that ADSL gene is very conserved in Drosophila. So the homology reached 60%, that is very high in terms of, let's say, if we talk about evolution, we are far, far away from the Drosophila, of course. But the moment that we have the same genes that literally conserve in more or less in the same way, and we figured out also that all the mutation that we observe in human, the same, let's say, sequence are conserved in Drosophila. This means that those amino acids, those, let's say, sequence of ADSL genes are very, very important because are conserved between Drosophila, C. elegans, and humans, mammals. So mean that we are talking about some, let's say, part of the ADSL gene and thus ADSL protein that are critical for the function of these genes. We play a little bit on this this C. elegans model. We downregulate ADSL because at that time we couldn't insert the mutation of ADSL. And based on this, we observed that also in this model, we could verify that there was a defect in mitochondrial morphology, activity, and so on. So this means that even if we try to change models, the moment that we touch ADSL, there is a defect in the mitochondria. I try to make the story short because otherwise it takes too much time. But one of the coolest point was that the moment that we try to downregulate, so we downregulate ADSL expression only in the muscle of these fruit flies, we observe a defect in mitochondria in the muscle, but we observe an alteration of motor neurons. So, and in motor neurons, we didn't affect ADSL expression. So this suggests that the moment that ADSL is downregulated in the muscle, this causes a wave of defect that can, let's say, affect also, negatively affect the motor neurons, and there is let's say, degeneration of what we call neuromuscular junction, okay? Then we play a little bit around. I don't give you too much detail because otherwise I guess it's gonna be too complicated, but we've tried to figure out how ADSL could be linked to the mitochondrial defect. So we check different kinases, so different proteins that are described to regulate purine synthesis, the non-purine synthesis pathway. We focus on our attention of AKT and ERK2 because they are being described directly to modify two different enzymes in order to boost or to activate the novel purine synthesis, okay? And we found out that these kinases are less activated in ADSL deficiency cells. And this is we use different way to stimulate these kinases. And in all the cases, there was a defect in the activation of these kinases, okay? So we tried to, because we couldn't, to be honest, at least at this point. So maybe in the next future, I hope that I'm gonna have further results. But we couldn't find a direct link between these kinases and ADSL. So we couldn't figure out how the cell could directly or indirectly regulate these kinases. We play the other way around. So we try to overexpress. So we try to activate ERK kinases in Drosophila, for example. We overexpress ERK. Why we overexpress ERK kinases? Because ERK kinases is being described to be involved in cancer. So there are a lot of specific inhibitor of these kinases, because when it's too much activated, is related to cancer in different organs. But so far there is no activator, or at least a specific activator for these kinases. So we had to overexpress the kinase per se, and we overexpress two different forms. One that is just the enzyme per se, the normal enzyme. And in the other hand, we overexpress the constitutive active form of these kinases. So sort of overboosted kinases, okay? And these kinases, the constitutive active form of these kinases could rescue the lethality in Drosophila that we observe when we downregulate ADSL. And specifically, we could observe that this kinase could partially rescue the mitochondrial defect observed in motor neuron when we downregulate ADSL. So meaning that this kinase could, let's say, at least partially rescue the mitochondrial defect. And we repeated this experiment in those cells derived from patients. Also in this case, we overexpress what we call ERK wild type, so the normal enzyme. To be honest, we overexpress ERK2 kinase because there are two forms, ERK1 and ERK2. But anyway, and in these cells, of course, the stronger results have been obtained. We obtained with the constitutive active form of ERK kinases. So we could partially rescue the mitochondrial defect in terms of mitochondrial number, amount, and also a little bit on the activity. But because, let's say, it's difficult to, as I said, to work with ERK, we try to use a different way to rescue the situation. We did metabolomic analysis. on fibroblast, on lymphoblastoid cells. And also in this case, we couldn't figure out a common defect between all the cell lines that we used. So look like that each single cell line could be something unrelated to the others. The only, let me use the only thing that we could observe was a tiny increase of S-ADO in the mild and severe cells, but this increase was not so huge as expected, okay? And we couldn't observe, for example, a huge increase of Sycar, and we just observe defect like or a little bit around, but there was not, let's say, a critical downregulation or a critical upregulation, okay? So we decide to, also in this case, play with the amount of adenine and guanine. Why? Because it's previously described that in some cases has been used adenine supplementation to rescue some mitochondrial defect. And say, okay, even if we don't see a strong decrease in adenine level, let's try to see if what happens if we boost adenine amount and guanine level. And as you can see, maybe from the picture, but from the graph, so we could rescue the morphology of this mitochondria. And okay, sorry. And we could also rescue the defect related to the mitochondrial DNA copy number. So meaning that probably if we boost the amount of adenine and guanine in these cells, we could rescue partially the defect observed so far in this cell line at the mitochondrial level. And we could rescue also the respiration in these cell lines. And so we obtain, let's say, a partial rescue that we publish, as I said, on the journal Cell Reports, but we was literally a fight. We spent almost more than 1 year to reply to all the comments of the referee and convince everyone that the story was solid. So I give you some unpublished, let's say, overview of what we are working now, because of course, the moment that we observe that there is a defect, mitochondrial defect, how we can, we need to figure out which is the link, okay? And so we are working on the fact that ADSL is localized on the mitochondrion. We don't know if it's inside the mitochondria or outside, and this make a big difference in terms of activity and function. Plus, we generate a mouse model in which we introduce this mutation that has been described in a mild patient in Rome, and we introduce this mutation in this mouse model, and we are still working try to characterize this mouse model, because it was literally, again, a nightmare, because at the first instance, normally the moment that you want to study the function of a gene, you downregulate the gene. So you eliminate the gene from the genome of the mouse and see what happens. Unfortunately, this is lethal. So we lost 3 years, try to find out a way to downregulate ADSL. So this is why now we introduce directly the mutation. And we select one mutation, because in order to create this mouse model for us cost a lot in terms of time and money. But the first result that we obtained is that also in the mouse model, the moment that we introduce this point mutation, we observe a decrease of the ADSL protein level. So it means that also in the mouse model, we observe what we describe and we observe in the cells derived from ADSL patients. So probably this model, even if let's say now we are characterizing the model different ages to see what's going on, not only in the first instance, so the first month of age, but also we are following the mouse development, and we are making a lot of results at the 9-month age, when mouse are very, no, not old, but adult, let's say, to figure out what's going on. And I hope the next time I can provide a lot of new data. So let me conclude because maybe I spent too much time. So now we are working to assess the ADSL function at the mitochondrion. We are characterizing this new mouse model, and we are developing new iPS cells, because another problem that we have the moment that we work with these immortalized pluripotent cells is that we need to get the proper control, and we are making isogenic cells. So it means that we rescue the mutation in this cell line that are in ADSL gene in order to see which is the, let's say, the effect on this, when we rescue specifically the mutation, and so we have proper controlled cells. Plus, we hope to get some new cell line where we have just single mutation in order to see how the single mutation can directly affect not only ADSL activity in terms of de novo purine synthesis, but also ADSL new function at the mitochondrion, I hope. And these are the acknowledgement. And as you can see, we are struggling a lot in order to get more funding from different opportunities. We won a grant from Novo Nordisk in Denmark, because we apply also in Denmark, because we had a lab in Denmark, and I spent almost 3 years in Copenhagen. We get some money from European community. We got a grant from Teleton, that is an Italian, let's say, foundation that is focusing on rare genetic disease. Now that we have the mouse model, we try to, let's say, get more attention. I hope to get more money to, again, to expand our research and, let's say, get better results and figure out what's going on in this mouse model properly. And thank you all for your attention.
That was great. Thank you so much, Dr. Bordi. I'll start with one. I want to acknowledge we're at 9:30, which is supposed to be the end. I'm hoping that most of you will be able to stay a further 15 or so minutes so we can get our Q&A with Dr. Bordi and wrap up. But if you need to leave, please know I understand. All right, Dr. Bordi. So Dr. Chen has asked in the comments, ADSL protein level in patient fibroblasts seems to correlate well with disease severity, but not ADSL mRNA. What is your thought on this result?
Exactly. This is true. It's something that, because normally when you see a decrease in the mRNA level, normally you do have the decrease in the protein level. The point is, there is no direct correlation. So, and we think that there is a further regulation that can explain this discrepancy. So now we are trying to figure out why. So we are working at the ribosomal level, for example, to see I don't want to, maybe it's a little bit complicated, but try to simplify. So the moment that the RNA is generated, need to be translated in amino acid sequence. And this is the job made by ribosomes. So it can be, if there is a mutation, that ribosomes are stuck on this mRNA. And this can cause a delay in the production of the protein or a defect, literally. So we are trying to figure out if this is the case, or probably there are other explanations, because we exclude that ADSL is more degraded, but we didn't exclude that is cleavage, for example, in different way, because when a protein is can have some alteration in terms of structure, there are other systems that can, let's say, make some quality control. And the moment that the protein cannot pass the quality control, normally is degraded and can be degraded in different way. So we are trying to see if there are other pathways that can be involved in this, let's say, sort of degradation, and can explain this discrepancy.
You talked about your mouse model. And I just want to say we have two gene therapy teams that are struggling to develop a mouse model right now. So I know you all have information to share with one another. What made you choose the R309H variant for your murine model? Was that just the most successful?
No, let's say that, okay, so we spent almost 3 years trying to, let's say, make the knockout. The moment that was for us very impossible, and we applied different strategies, and I can share everything with the rest of the community, scientific community on this, and I already did a little bit. We pick up one mutation that was related to mild disease because our idea was if we pick up a mutation that is related to the severe or fatal neonatal, maybe we could have the same problem. So let's try to, let's say, simplify a little bit and try to get an animal first. Then the moment that we are able to get an animal, even if it's not the most common mutation and is not like something that should, I mean, in fact, we don't have a lot of, let's say, beautiful result as expected, but we have a lot of results.
Yeah.
We try to, we are still working on try to understand what's going on properly, but this was the case. So spending three years to try to generate a mouse model, and we try to, let's say, pick up the mutation that was, let's say, less severe. Let me see
Yes.
this.
Yes. Dr. Chen asked, are you willing to share your mouse model if you see behavioral and molecular phenotypes?
Sure. I mean, it's just so we are here to share everything. I talk with most of the people, my colleague here. We are in contact. Everyone here is very nice in terms of we reply to each other very quickly, and it's not very common because sometimes it's something that we don't see around, or at least so commonly.
Yes, it's wonderful. I mean, obviously, as a patient group, we really appreciate that kind of working together in collaboration because we know that helps our kids. So we thank you for that. I wanted to ask, oh, okay. Wendy, I saw you pop on. Do you have a question?
Yeah, I was going to ask questions. Is that all right?
Yep, of course.
Uh, very, very nice, Matteo. Thanks. That's such cool work.
Thank you.
I am curious what your thoughts are. You know, the purine nucleotide cycle um, is known to have interactions with what's going on with mitochondrial metabolism, and in a number of studies, supplying fumarate can really be helpful. And I wondered if you just tried that.
Yes. So let's say that now we move our attention, to be honest, on Krebs cycle,
Yeah.
because we did the other, let's say, bioinformatic analysis. So I did include all this data, but if you want, we can, let's say,
Mhm.
meet
Yeah.
uh, next days and I show you all the other molecular details that we have now, because we know that ADSL is localized at the mitochondria, and he's able to interact with different proteins at the mitochondrial level. And when we did some other bioinformatic analysis, we connect ADSL to Krebs cycle. So Krebs cycle, I explained to everyone. So in order to obtain ATP, so means in order to store energy, the cell need to do glycolysis, Krebs cycle, and oxidative phosphorylation. So all these three metabolism are connected to each other, and they can interact to each other because there are a lot of steps that are tiny regulated. So we think that ADSL is involved somehow. To be honest, I don't know if is directly related to the fact that ADSL can produce fumarate or is able to do something else, a sort of moon-like protein. Depends where it is. So in the cytosol can be mostly related on the novel purine synthesis. In the mitochondrion, I have some hypotheses, but I'm still trying to struggling to see if these hypotheses are, good or not. Let's say this.
Thanks.
Anybody else with a question? I have another question. So
Yeah, sure.
yeah, you spoke a little bit about early development. So we've talked twice. We've had two talks today that kind of talk about an ADSL patient in utero, and I just want to clarify or ask your thoughts on even if a child, if we get a treatment, a perfect treatment, and a
Mhm.
child is born and they receive that treatment, we are still going to see effects because they've developed in utero with ADSL deficiency. Is this something we can say, or do you know?
Uh, I mean, I don't have the right answer, okay? Because there are a lot of, I mean, so many aspects that we still don't know, but not just related to ADSL deficiency, are related also to nervous system development per se. So it's so complicated that it's difficult to figure out which is the best window in order to get, let's say, a treatment,
Mhm.
okay? Because at the moment that we are able to, let's say, make a link between the function of a specific gene with the development of nervous system, and then there are a lot of different aspects that we need to consider in terms of, okay, it's true that at the end, I mean, the end of the process of the nervous system, I mean, the process of development
Mhm.
ends let's say, very late in life. Okay. There are a lot of neurons that are generated months after the birth. So the nervous system is a beautiful machine, but if we don't, let's say, figure out how ADSL is involved in this process at the molecular way, then it's hard to make hypotheses that can, let's say, revert or at least reduce the impact or let's say ameliorate the defect that we observed in the nervous system.
Mm. Yes.
I'm sorry if I cannot respond better of this, but it's also very difficult, complicated because we are trying to, let's say, make some behavioral tests on this mouse model, okay? And we struggling a lot try to figure out which can be, let's say, the best behavioral test in order to get a defect.
Mhm.
And the moment that we have the defect, we need to go back and see, okay, why that, let's say, alteration is occurring only when we do that specific test and not when we do another test. Okay, and then we move to the molecular level, and
Yes.
we try to catch the rabbit in the hole.
Yeah. Wendy has
Yeah,
something.
I just wanted to respond to your question as well. I think that you're going to find both developmental and acute function of ADSL playing a role, I think, right? So you're right. You can never go back and make sure ADSL is there from the very start, and that's likely to have effects, right? But I also know that, I mean, like we've thought about this a little bit in worms, you can allow the animal to grow up to an adult and then remove ADSL function and things go wrong.
Mhm.
Right. Or you can allow the animal to grow up without ADSL and then put ADSL back and you can fix certain things.
Yeah.
Right. And so there's going to be both an acute need for ADSL and there's going to be developmental effects of having not had ADSL. There's going to be both.
Mhm.
And it's going to be a while before we know, I guess, how if it's possible to get over the developmental ones at all,
Yeah.
right?
Yeah. Especially because I've been talking with Dr. Oleg a lot about myelin, the changes in the myelin. Can myelin be rebuilt? I think that I've been following some people that study MS, multiple sclerosis, and it's interesting these diseases that have these correlations. Like we've talked several times about Parkinson's. MS seems to have some crossing ideas as well. So I'm very curious about that. going forward, when we come up with a treatment, what will we see? It's very interesting to me. Do we have any other questions for Dr. Bordi or anybody else here? No? Wonderful. I want to say thank you all so much for being here today and for going a little over the time. Um, actually, not so bad considering how far behind we started. So I'm going to wrap up and say I will be sending out, it's going to take me a little bit to get this recording divided up, but the recording will be available to everybody to look back and continue to learn from, to ask more questions about. And we will see you next year, if not before. Thank you all so much.